Essay number 7 is not covered on this lecture exam. Essay number 7 is no longer on this exam. So that's a third of the material out of this exam. So you guys should be able to do a lot better, okay? So no sensory systems on this exam. It will be covered on the final. It will be covered on the final. So essay number 7 will become essay number 1 for your final exam list, okay? So add this to the final exam list. Is everybody clear about that instruction? Yes? Okay. So let's do all the let's do all these other essays. Again, take good notes, okay? Because I'm going to be going fast.
So just 1 through 6 on this one.
It's only 1 through 6. And brain table. Don't forget to do the brain table. Brain table's on this exam, lecture exam as well. Okay? I could easily give you a 20-point table, okay, on the exam. So please make sure you know your brain table. I will not ask location on the lab exam or the lecture exam. No location. Are we clear? Location's just there to help you. Okay, the location part of that table, you're just using that as a guide. I'm not going to ask you to memorize that under any circumstance. 1. Describe the synaptic events that cause the neurotransmitter exocytosis and lead to IPSP and EPSP at the synapses. Well, first of all, action potential comes down the axon, causing sodium to come in. When sodium comes in, it causes calcium channels to open. Calcium comes in, causing the vesicles to fuse and release neurotransmitters into the synaptic space. Okay, that's what happens with exocytosis. All right, so how does it lead to EPSP and IPSP? So you need to know what those are. EPSP are excitatory postsynaptic potentials. IPSPs are inhibitory postsynaptic potentials. Excitatory means you will cause a depolarization and possibly an action potential. IPSP means you cause hyperpolarization. Okay, so depending on the neurotransmitter receptor, on the postsynaptic membrane, you can have either an excitatory or an inhibitory potential. Describe the functional regions of neurons and use this to explain the presynaptic position. So there are 3 important regions in the neuron. You have the cell body. The cell body consists of dendrites, which are receptive antennae, and you have mostly non-voltage, not mostly, it's all non-voltage gated channels. So it can be chemically gated channels, it can be mechanically gated channels, it could be pretty much any kind other than voltage. And that's what you find in the cell body, including the dendrites. That leads to graded potentials. Graded potentials always reflect the stimulus strength. Okay, so you have a weak stimulus, you have a weak graded potential. You have a strong stimulus, you have a strong graded potential. And all of these signals are then summated or integrated at the axon hillock. Axon hillock is where you find voltage-gated ion channels first. The first voltage-gated ion channel happens to be located in the axon hillock. Excitatory signals can come from the cell body, and if they're strong enough, they can reach the axon hillock. The signal won't be lost. Inhibitory signals are weak to begin with, and so if you put them really far away, they could be lost. So inhibitory synapses are found very close to the axon hillock. And on this one, you have to explain it. You have to explain why excitatory is strong and why inhibitory is weak. I've gone over this in lecture, so go over that. I'm not rehashing everything from lecture right now in half an hour. So check your lecture notes. It's all there. And lastly, explain the mechanisms of spatial and temporal summation. Remember that spatial summation deals with simultaneous release of neurotransmitters, simultaneous release of various neurotransmitters, and temporal summation, it's only one neurotransmitter being released at a certain frequency. Spatial summation can be excitatory or inhibitory. Temporal summation can only be inhibitory. Excitatory. Temporal summation cannot be inhibitory. Temporal summation can only be excitatory. Any general questions on number 1? Explain how current spreads down the axon and triggers subsequent action potential. Compare and contrast this propagation in myelinated versus unmyelinated axons. Include in your discussion the distribution of voltage-gated channels, why those differences are necessary. Explain why white matter is largely myelinated axons, while axons in gray matter are usually unmyelinated. Current spreads down the axon because of voltage-gated channels. Okay, in unmyelinated axons, voltage-gated channels are distributed uniformly, just like in skeletal muscle. Remember, it's like a commuter train. It has to stop at every station. So it takes a long time. In unmyelinated axons, it only travels at 3 meters per second. And in myelinated axons, you have a large layer of myelin, okay? And ion channels are only concentrated in the nodes. Ion channels are only concentrated in the nodes. So action potentials tend to jump from one node to the next. And that's called saltatory conduction. And why is this necessary? Well, ions cannot go through myelin. So no point in having ion channels underneath the myelin. Okay? You also have to explain that in both cases, the signal will lose strength. The signal loses strength faster in unmyelinated than it does in myelinated axons. And every time you hit a node or a voltage-gated channel, the signal becomes replenished. We went over all this. Signal gains strength at every place where you have a voltage-gated channel. Okay. So explain why white matter is largely myelinated. Well, white matter it can be classified into 3. Right? Tracks that travel long distances on one side of the brain. Commissures, which cross the hemispheres of the brain, so they're kind of short. And association fibers, which are really short. And those are within the same side of the brain. They're very short fibers. When you have very short fibers, you don't need myelin because short fibers are used for processing. And when you're using a structure for processing, it has to be done carefully and slowly. So you don't want myelination there. Questions on 2?
Do you send the commercial for
I don't think they are. Yes, they could be. I mean, the corpus callosum is myelinated, so yeah, I think they could be. You guys have done number 3 already. The table of neurotransmitters. It's already there in your PowerPoint. Basically, I need you to in the table basically lists the name of the neurotransmitter, what class it belongs to. There are 6 classes. The first class is acetylcholine. The second class is biogenic amines. The third class is going to be peptides, okay? And 4 through 6, you're dealing with purines, gases, yes, and blanking on the 6th one. Amino acids, thank you. Amino acids. So you need to know at least one example for each, and you should know whether they're excitatory or inhibitory. You should also know if they work by a direct mechanism or an indirect mechanism. Direct mechanism is when a neurotransmitter opens ion channels directly. Indirect mechanism is when a neurotransmitter receptor activates an enzyme, and that enzyme makes cyclic nucleotides like cyclic AMP, and then cyclic AMP opens ion channels. So it's very indirect. It takes a long time. So that should be in the table. Explain how the same neurotransmitter can excite one target and inhibit a different target. Well, the same neurotransmitter can activate very different kinds of receptors, and you should use epinephrine as an example. Epinephrine has 5 classes of receptors, right? And in one tissue, epinephrine can cause muscle contraction, another tissue it can cause the muscle to relax. Because the receptors are very different. Okay, that's how a neurotransmitter can have a variety of effects, is because it binds to a variety of neurotransmitters. And remember, you have to explain it. Okay. And the indirect mechanism, I need to see all the details. The neurotransmitter binds the receptor. Then your G protein becomes activated. The G protein will activate adenylate cyclase. You can always just say enzyme. Okay, it activates the enzyme that makes cyclic AMP. Cyclic AMP is called a second messenger. Then cyclic AMP will then have all of its effects, including opening calcium channels or any ion channels. Okay, then you compare contrast direct with the indirect. Questions on 3? Four, describe 3 organizing principles of cortical structures: cortical mapping, lateralization, and relationship of primary versus associative functional areas. Okay, so cortical mapping, there are 2 aspects here. One, all the organs in the body, okay, the areas corresponding to these organs also appear in the same order. This is called mirroring. You mirror how the organs appear in your body, you mirror that in the brain. The areas are distorted, meaning the areas are unequal. It really depends on how much you use a particular area. The more you use it, the more skilled a particular area is in the body, the more representation it's going to receive in the brain. Lateralization describes the uniqueness of hemispheres. Don't just write that and leave it. You have to give me an example for each of these things. Lateralization, Broca's area, Wernicke's area are examples of lateralization. They're only found in the left hemisphere. They're not found in the right hemisphere. Plasticity. Plasticity is how you can change the area, the cortical area, through practice and learning. And lastly, contralateral is the fifth principle. Contralateral basically deals with the right side of the body is represented in the left side of the brain. The left side of the body is represented by the right side of the brain, controlled by the right side of the brain. So you need to explain that. And this has to do with decussation of nerves. Nerves decussate often and go to the opposite side. To explain motor unit sizes and motor control, well, the motor cortex is also mapped like this, and the larger the area, the cortical area, the more precisely controlled the muscle is going to be. Okay? Use the concept of sensory receptive fields to explain the reasons that distortions also occur in the sensory homunculus. Okay? We did this in last class. We're going to do it in lab today. If the receptive fields are going to be really big, okay, big receptive fields usually means that your acuity is going to be high or low? Low. Your acuity is going to be very low. If you have a large area that you have to control, you're going to be less accurate. Okay? And if the receptive field is really high, what's going to be, or large, what's going to be your cortical representation?
Small.
Small. Okay, good. Okay, so you need to explain that. That's it. Essay 4. Number 5, describe the arrangement of motor and sensory functions in the spinal cord. Describe the 3 ascending pathways between or within the spinal cord. Include the general functions carried out by each of these ascending tracts. Describe how motor information descends along pyramidal and extrapyramidal tracts and contrast the respective functions of these tracts. They're all independent questions. Motor and sensory functions. Let's show you. In the spinal cord, you always start out by saying that the spinal cord has white matter on the outside and gray matter on the inside. The gray matter is arranged as a horn. The dorsal side of the horns, or the dorsal horns, are sensory in nature. The ventral horns are motor in nature. Extreme tip of both of those are somato, meaning they go to voluntary muscles, voluntary organs. The mid portion, okay, so mid-sensory and mid-motor region are always visceral. Visceral means your heart, your kidney, your intestine, liver, those kinds of internal structures. So you have to describe those. And then it says describe the 3 ascending pathways. You already have the map. Just draw the map. If you want to write it, you can always talk about the first order neuron, the second order neuron, and the third order neuron. Okay? And describe where any of them decussate, if at all, and how they move from the spinal cord to the brain. Pyramidal tracts come down from the cerebrum, from the primary motor cortex, and they basically deal with somatic motor pathways, and they're usually inhibitory. We talked about this last lecture. They're typically inhibitory, okay? And we we have proof about this from Clonus. Okay. Clonus. The descending. Extrapyramidal starts in the red nucleus in the medulla, and it's primarily involved in involuntary movements like balance, posture, standing, sitting, that kind of thing. And you should explain that most descending tracts are inhibitory. Okay? And explain why using the bonus. Okay. That's pretty much it, okay, for this essay. It's a short essay, actually, number 5. Don't forget, you still need to know all the neuroglia, the CNS and the PNS neuroglia. There's no essay on it. You need to know those. You don't forget that. Write that down. And you need to know the brain table. The last essay described the role of 5 components of general spinal reflexes. The 5 components, you have to define each one of them. Don't just name them. You need to give me the definition of each one of those. What do they do? What are they? The receptor senses the change. The afferent pathway conveys the change to the spinal cord. Its cell bodies in the dorsal root ganglion. The spinal cord could use interneurons, but the spinal cord basically deals with a very simple response through the efferent pathway. And then the efferent pathway goes into skeletal muscle. Usually, if you're talking about somatic reflexes, or it goes into smooth muscle if you're talking about visceral reflexes, or cardiac muscle for that matter. So it says apply these principles to the mechanism of patellar stretch reflex. So you basically have to say that if the patellar region is activated by a hammer, then the muscle spindle in your quadriceps starts conveying that action potential to the brain. I'm sorry, to the spinal cord through the annulospiral neuron. Through the annulospiral neuron. Annulospiral sensory neuron hugs the muscle spindle. What happens? What's the other sensory neuron here? Flower spray, okay? And that's found at the ends of the muscle spindle. So when the muscle spindle contracts to reset itself, the flower spray will continue to excite the alpha motor neuron, so that during those few seconds, the alpha motor neuron doesn't stop. Okay? Typically, the alpha motor neuron does 2 things. It activates the prime mover, contracts the prime mover, but it also relaxes the antagonist. It relaxes the antagonist. The alpha motor neuron. So it asks you to discuss reciprocal inhibition. Reciprocal inhibition is one where you inhibit the antagonist, but you enhance the prime mover. Explain why alpha gamma coactivation is necessary during voluntary movements. Again, we did this last class. Alpha must continue to get excited for extrafusal fibers to be contracting, and gamma will cause the resetting of the muscle spindle. And if the muscle spindle doesn't reset, the contraction ends there because there's no signal sent to the spinal cord. So gamma motor neuron, it only senses a contraction or causes contraction of the muscle spindle. Okay? Questions on 6? Remember, reciprocal activation is not on your test. I'm not going to ask you that. Perfect timing. Questions? Okay. All right. Well, take a 5-minute break.